Hello everyone and welcome. I'm Roisin from Wiley and I'll be your moderator today. Thank you for joining us for this webinar which is sponsored by SciTech. This presentation explores how lung epithelial cells communicate with CD4 plus T cells to regulate immune function and location during bacterial inhalation and what this means for the pathogenesis of neutrophilic asthma. Our speaker today brings exceptional expertise. Dr. Anukul Shenoy is an assistant professor in the Department of Microbiology and Immunology and the Division of Pulmonary and Critical Care Medicine at the University of Michigan Medical School. So just before we welcome him on, I would like to let you know that this broadcast will be recorded and made available for on-demand viewing. We encourage you to use the chat during this presentation to ask any questions you may have, which I will then address during the Q&A session afterwards. So now, without further ado, I will turn the floor over to you, Professor Shenoy.
Okay, so thanks for the generous introduction. Today I'll be sharing a saga about what got us interested in studying epithelial cells and their interactions with CD4 T cells in context of lung immunity. But a lot of this work has cross-applicable impacts and implications on other barrier tissues as well. So we have been very interested in understanding the epithelial cells and their biology in different barrier tissues like the conducting airways, the alveoli, the intestine, the skin, the nasal epithelium. You think of any barrier tissue, the first cell type you think of as acting as a direct interface between the core of the body and the external environment is our epithelial cells. From the perspective of an epithelial cell, it is constantly being exposed to a bunch of different environmental insults. These include different types of microbes, some of which are commensals, many of which are opportunistic pathogens, different types of environmental insults like smoke, cold air, like right now in Michigan, different types of pollen particulates. And these epithelial cells that are constantly getting bombarded with all these different environmental insults have to make sense of all this information and decide when it needs to launch and activate the immune response so that the immune cells can come into the tissue and help clear these insults. Another interesting thing the epithelial cells does is once that inflammation is cleared, the epithelial cell also is in charge of essentially acting as a stem cell in that organ and helping with tissue repair, which again has to be done in close concert and communication with the immune system, because if this fails, this is a recipe for cancer. So we have been very interested in understanding how is it that this epithelial cell that essentially sees everything we see in our environment is constantly integrating all these signals, making sense of this, communicating with the immune system so that most of us can have a long, healthy life. And one of the immune cell that we got interested in looking at in context of epithelial communication is a type of a newly discovered T cell subset called as the tissue-resident memory T cells. So tissue-resident memory T cells are a unique subset of memory T cells that are present in multiple different tissues, and every few weeks or months, there are new papers showing presence of tissue-resident memory T cells in multiple different organs. What sets these tissue-resident memory T cells, or I will call them TRMs to make things easier from here on, apart is that compared to the other memory T cells, like the central memory T cells that move from the blood to the secondary lymphoid organ and back to the blood, or the effector memory T cells, which go from the blood to the tissues to go back into the lymphoid organ and go back to the blood, the tissue-resident memory T cells, once they have been seeded in their non-lymphoid tissue, they essentially stay put. And they essentially are thought to replenish themselves by in situ proliferation. These T cells squiggle around in the tissue just under the epithelial layer and essentially, as I mentioned, can undergo proliferation to maintain their numbers in the tissue. But what they are usually doing is they're acting as adaptive immune sentinels in that tissue, looking for the pathogen or a cognate antigen that might show up in that tissue. Once it does, this T cell now, because it is there at the front line, gets activated quickly to produce different types of cytokines and chemokines to call for other effector T cells. And if it is a CD8 resident memory T cell, it can actually just kill the infected cell to essentially stop, for example, a virus infection before it festers and infects more cells around it. So I've been talking about epithelial cells in a tissue, and I've been talking about it as if they are this one big umbrella cell type. In some ways, yes, but not all epithelial cells are the same type. So, for example, using the lung as our model barrier tissue, one thing that we can see is different parts of the lung, such as the airways versus the alveoli, have different types of epithelial cells that occupy that niche. For example, if you look at the airway up here in the trachea and the bronchi, you will see that there are these cells called as club cells or secretory cells. And these cells do a job of secreting antioxidants and multiple different detoxifying enzymes that essentially make sure the air that we breathe is detoxified. There are also these ciliated cells that have these hair-like structures on top of their head, if you will, and these cilia are constantly beating in an upward direction to essentially throw out all the particulates that are trapped in the lung. There are also a variant of club cells or a differentiated form of club cells called mucus cells or goblet cells that produce mucus, and that is the mucus that we usually cough out when we are having a cough or an infection. So these epithelial cells in the airways are trying their best to make sure that none of the things that we breathe in that might cause us harm ends up down in the alveolus. And why is this important? Well, the alveolus is a less than micron thin, and its main job in the lung is to oxygenate our blood. So when these alveoli are oxygenating our blood, any sort of an infection and inflammation down in the alveolus can lead to a problem, and it's not good for survival. So this layer of the respiratory epithelium is formed by a subset of epithelial cell called an alveolar type 1 cell. And then there is another subset of epithelial cells, which we will talk a lot about, called the alveolar type 2 cell. Now, this cell is really interesting. Its day job is to produce a bunch of surfactants, which essentially allows our lung to expand and contract all the time as we are breathing without ripping our cells apart. And its night job is if there is some sort of an injury that happens in the lung, these alveolar type 2 cells act as stem cells in the lungs to differentiate into alveolar type 1 cells to heal this injury. So we got interested in asking, how are these different subsets of epithelial cells talking to CD4 T cells in the lungs? And like any research project, we needed some sort of a perturbation system to ask this question. And one obvious question for us to ask was the disease called pneumonia. So for most of the audience, having come out of a COVID-19 pneumonia pandemic, this does not need an introduction. But just as a refresher, pneumonia is essentially an infection of the lungs, which causes a lot of immune cell and vascular leakage into the airways, so that the airways, which were meant for breathing, cannot do the job of breathing anymore because it is filled with all this fluid and inflammatory cells, as a result of which it causes difficulty in breathing and death. It is the leading cause of infectious deaths in the elderly, and by that extension, also a large socioeconomic burden worldwide. One thing that is very interesting about most of the agents that cause pneumonia is they are actually caused by ubiquitously present opportunistic pathogens. Many of us carry it on our nose all the time. And in very young children who have an underdeveloped immune system or very elderly because of immunosenescence, these people tend to suffer from a lot of cases of pneumonia. Surprisingly and interestingly, it's the elderly individuals that succumb to pneumonia, whereas in young children, the rate of mortality is not that high. And then there is this really nice period of our life as young, healthy adults that we appear to possess immunity to most of these pathogens. So our lab, where I was doing my postdoc with Jay Misgard, was interested in asking what is going wrong in these older adults. And one way for us to think about this is if we want to know how a clock, what caused the clock to break, it's useful to know what allows the clock to work. So in other words, to understand what is going wrong in the aging adults, maybe we want to first know what is going right in the young adults. And the hypothesis we decided to first follow was this hypothesis that these early life microbial exposures that kids are having when they, as soon as they're born and growing up into their adolescence, they are seeing all these different bugs for the first time in their life. And many of these exposures might be important for developing an immune response in the lung that protects us as young adults. So how do we ask this question in the lab? So for this, we use a bacteria that I did my graduate school work on, which is called Streptococcus pneumoniae. Streptococcus pneumoniae, as you can see in this electron micrograph, is a diplococcus that is gram positive, and it also expresses this layer of sugar capsule on its surface. This bacteria is a frequent nasopharyngeal colonizer. Up to 60 to 80% of young children have it in their nose. Up to 40 to 50% of young adults carry it with them in their nose, and we are breathing it in all the time. Most often than not, in an healthy individual, it does not cause any sort of a disease, but if there is severe immunocompromise, this bug can cause disease. So what we do is we essentially take C57 mice, C57 SPF mice, expose them to non-lethal isolates of Streptococcus pneumoniae 1 week apart, essentially mimicking these recurrent early life exposures the kids might be having to these bugs. And then we let the mice rest for 4 weeks, essentially allowing the human to grow into young adulthood, if you will. And then we challenge the mouse with a lethal strain of the same bacteria. And what we are finding here is in an experienced lung, which we also called as a heterotypic immune mouse, we see a robust influx of neutrophils within 7 hours compared to a mouse that has never seen a bacteria before. And what is very interesting is even though the number of neutrophils by 24 hours have equalized out, this early neutrophil influx is what allows these experienced lungs that have different combinations of experiences to clear the bacterium. So a faster neutrophil recruitment is what translates to better protection. And number two, that translates as better survival for an experienced mouse as well. Mechanistically, what we found as the main driver of this was a neat experiment that was run by Dr. Nicole Smith in Jay Misgard's lab, where she gave exposure to the mouse to the non-lethal isolates only in the left lung of the mouse. So essentially the right lung of the exact same mouse acted as the control, and both the lungs technically are sharing the same circulation. And what we find is only the left lung of that experienced mice have developed this subset of CD4 T cells that are positive for CD11A and CD69, which are really good markers for tissue resident memory T cells. And the right lung of that exact same mouse does not have that subset, suggesting it's a very regionally compartmentalized immune response, even in the same mouse, if you will. And if we challenge both these lungs in the mouse, what we find is the left lung, which has these T cells, are able to clear the bacteria much better than the right lung, which does not have these T cells. Again, suggesting that these CD4 resident memory TRM cells are what is the major driver of the protection. So consistent with the fact that we saw a robust neutrophil, we had a good clue that these might be Th17 resident memory T cells in the lungs. But we again went to confirm the same output by showing that these T cells from an experienced lung on re-stimulation are capable of producing a lot of IL-17, which other literature have associated with robust neutrophilic inflammation in the tissues as well. Most importantly, if we give GK1.5 to this mice, and what GK1.5 does is it depletes the CD4 T cells from the mouse, we could show that despite the experience, the mouse that has gotten GK1.5 actually loses its ability to control the bacteria, suggesting that it is these CD4 T cells that are driving this protection. So far, what I have told you is this microbial exposure induces this seeding of the lung with this bunch of CD4 resident memory T cells that are producing IL-17 that somehow activate the neutrophils to come into the lung and fight the infection. But the big question that we essentially tackled in this manuscript that is published was, how are these Th17 T cells calling the neutrophils in the lungs? And one question we asked was, we know neutrophils require chemokines to follow the chemokine gradient to get into whatever tissue they are being called to. So we asked a very simple question. Number one, if we take an experienced mouse, and if we deplete the CD4 T cells and then challenge them, in absence of the CD4 T cells, number one, would the number of neutrophils go down? And number two, if yes, what chemokine might be correlating with the loss of neutrophil influx in the lung? Turns out, we found that depletion of the CD4 T cells reduced the neutrophilic influx in the lung, as we expected. But among all the chemokines that we know of driving neutrophil influx, the main chemokine that we saw that was reduced was this chemokine called CXCL5, or some people call it LIX, or also called as ENA78. If you do the correlation analysis for each of these data points, you see this really nice correlation where if a mouse has high amounts of CXCL5, it has called for more amounts of neutrophils. If the mouse has less amount of CXCL5, it has called for less neutrophils. Indeed, suggesting that the Th17 cells were somehow activating CXCL5 to call the neutrophils in the lungs. This discovery of CXCL5 to us was a really good indication and a guide, because prior work in the Misgard lab had shown that CXCL5 among all the cells in the lung is predominantly an epithelial derived chemokine. So that led us to ask the question of, could it be that the Th17 cells need to talk to the epithelial cells to induce CXCL5 production to call neutrophils in the lungs? So to ask this question, we took the same experimental setup, took naive mice or experienced mice, gave them the pneumococcal challenge, and then processed the lungs to sort out the CD45 negative EpCAM positive epithelial cells, isolated RNA, and did QRT-PCR for three main chemokines. One of them being CXCL5, but GM-CSF and CCL20 were the other two. The reason why we looked at these two chemokines as well is because prior work in our lab had shown that all these three chemokines are technically under the control of the same transcription factor, NF-kappa B rel A. Surprisingly, and also very interestingly, what we find is an experienced epithelium was able to produce a lot more CXCL5 compared to a naive epithelium. And this is very important conceptually. What we are saying is the same epithelial cells will produce very different levels of the chemokine to the same exact pathogen, depending on the fact whether they have had experience or not have had experience. And then the other really interesting thing was this did not seem like an overt activation of the epithelium, because the other two chemokines, GM-CSF and CCL20, were unaffected. So this seemed like a very selective activation of the lung epithelium. When we dug into what might be driving this selective activation, we found this really neat pathway that IL-17 activates, where once it signals into the nucleus, it induces transcription and translation of these proteins called AU binding proteins. One of the classical well-known example of an AU binding protein is HUR. And these AU binding proteins go and bind to the AU repeats that are at the 3' end of the mRNA molecule. So what this does is essentially prevents that mRNA from getting degraded. In other words, the cells do not need to transcribe more of this molecule or this mRNA, but the same RNA can be stable for longer, allowing for more protein to be synthesized from that mRNA. So to ask this, whether this was at play in our model, what we did is we took lung epithelial cells in a dish and stimulated them with TNF or TNF plus IL-17. And consistently, what we find is IL-17 indeed stabilizes the CXCL5 for much longer compared to TNF, where the mRNA for CXCL5 degrades very quickly. And secondly, because this is a very RNA targeted effect, we find this effect does not occur for GM-CSF and CCL20, which I showed you in the previous data plots, are not changed. So the selective activation is indeed by post-transcriptional stabilization. So one of the things an astute scientist would ask is, okay, you have been saying CXCL5 is driving neutrophilic influx, but you have not given me any explicit evidence other than a correlation plot to show that. So what we did here, which I think is a really neat experiment, is we gave the mice microbial exposure and then neutralized only the CXCL5. So what this experiment is telling you is the T cells in these lungs are completely intact, and all we are neutralizing is the epithelial-derived chemokine. And consistent with the CXCL5 neutralization, we also again saw the loss of neutrophil, which suggests that, you know what, you can have all the CD4 T cells under the sun in your lungs, but if your epithelial cells are not doing their job, those T cells are not going to be protective. And we can prove that by doing another experiment where if we take these experienced mice, give them GK1.5 to deplete the CD4 T cells, yes, the neutrophils go down. But even after depleting the CD4 T cells, if we give the epithelial-derived CXCL5, the neutrophil influx can come back up, suggesting that actually the epithelial cell plays as a very important effector in calling the neutrophils into the lungs. So hopefully from this story, I have kind of started to paint a picture where these epithelial cell and T cell interactions are actually very intimate, in the fact that the Th17 T cells via IL-17 can induce the epithelial cell to stabilize specific mRNA molecules, which allows the epithelial cells to produce more CXCL5, call neutrophils, and help us fight the infection. But I've never been a fan of a one-sided story. I always like to look at the story from both the angles. And this led me to ask the question of, if there are so many T cells in the lungs that are lined up along and with the epithelial cells, could the epithelial cells be telling the T cells what they need to be doing in the first place? And one of the biggest reasons why I went down this interesting direction was essentially asking or finding that these alveolar type 2 cells, which I mentioned are the main surfactant producing cells, they have for multiple decades been known to express a large amount of MHC class 2 in rats, in humans, in mice. It's very evolutionarily conserved. But the role for epithelial MHC class 2 in the lungs was not necessarily explicitly tested completely. And why am I really interested in asking this MHC class 2 question? Well, it's because of its most important role in essentially communicating from the innate immune response to the adaptive immune response as part of antigen presentation. So for the non-immunologists in the audience, I'll just give you a brief spiel on antigen presentation. So our professional antigen presenting cells, like a dendritic cell, a macrophage, a B cell, these cells have to eat up exogenous antigens like a pathogen, break it into small peptides, and put these peptides on the surface of a molecule, MHC class 2 here, to toss it on the surface. And this MHC class 2 with the pathogenic peptide on its surface plays a very, very, very important role in essentially translating the innate immune response into an adaptive immune response. And the way it does is the MHC and the peptide that I showed you from the last slide acts as signal 1. And there is a signal 2, which can be a co-stimulatory molecule or an inhibitory molecule. And only then will a T cell be able to recognize this particular antigen to essentially cause appropriate functions. So if you have a co-stimulatory molecule like CD4 T, 80, 86, or ICOS-L, these T cells are getting a positive signal, which drives the T cells to proliferate, start producing cytokines to differentiate, and essentially help us fight these infections. On the flip side, if these signals are inhibitory signals like PDL1 or PDL2, this antigen presenting cell can tell the T cell to essentially quiet down. It inhibits the T cell proliferation, causes the T cell to die, and may cause the T cell to become a regulatory T cell. And many of us are familiar with the molecule PDL1 because of its pro-tumorigenic role, where cancer cells express PDL1 to shut down T cells that come to kill it. So this antigen presenting cell has a huge onus on it. It is supposed to translate all this information that it got from the end of the innate immune response to the adaptive immune response in context of MHC class 2 and the co-stimulatory molecule, and also produce the right kind of cytokines, which will convert the T cell to become a T helper 1 or a 2 or a 17 or a regulatory T cell, depending on the needs of the system. So for example, in our model, I mentioned that our bacteria is an extracellular pathogen and requires IL-17 for the protection. So the antigen presenting cell has to tell the T cell via these cytokines to express ROR gamma T and become a T helper 17 cell. So given such an important role for MHC class 2 in antigen presentation, and the fact that epithelial cells are constitutively expressing MHC class 2, at least based on prior literature, it felt that we shouldn't have to ask this question as to why are epithelial cells expressing MHC in the lungs. So to tackle this issue, the first thing we did was ask, let's go back and see if we can find MHC class 2 on epithelial cells in the lungs. So here we used a mouse that expresses GFP downstream of the human surfactant protein C promoter. And remember, the surfactant protein C is expressed only in the alveolar type 2 cells. And what we find is all subsets of epithelial cells in the lungs, whether it be a club cell, a ciliated cell, and type 1 or type variants of type 2 cells, all of them are capable of expressing MHC class 2 in context of an infection. Now, you can have all the MHC under the sun, but that doesn't mean you can actually present an antigen to the T cell. So to test if the epithelial cells truly are antigen presenting cells to the T cells, we essentially created a mouse where we knocked out MHC class 2 specifically in the lung epithelial cells, sorted the epithelial cells out of the lungs, co-cultured them with CD4 T cells in the same dish, and essentially asked, now can the epithelial cell activate the T cell in a simplistic tube? And consistently, again, we find that if we delete epithelial MHC class 2, the T cells are not activated. And if it has MHC class 2, the T cells do get activated by the virtue of the fact that they upregulate CD69, which is an activation marker for T cells. So with this data, we had a proof of concept that the epithelial cells are indeed able to present antigens to CD4 T cells. But what does this mean for a tissue like a lung, which is constantly seeing a bunch of environmental insults, right? So the question then becomes is what is the function of the epithelial antigen presentation? So to ask this question, what we now do is we take the same mice I mentioned to you a couple of slides back, where we can knock out MHC only in the lung epithelial cells, delete the MHC using tamoxifen. After a washout, give the mouse the same experience to induce TRMs in the lungs. And then at day 56, we performed careful microscopic analysis to see where these T cells are in these lungs. And to our surprise, one of the really interesting things we found was most of the CD4 T cells like to be around the bronchovascular bundle and along the airways in the lung. They don't like to be around the alveolus. And furthermore, if you delete the epithelial ability to present antigen to the T cell, the numbers of these T cells around the airways and the bronchovascular interstitium actually fall down, which suggests that the epithelial antigen presentation plays a very important role in telling the T cells where to go in the lung, right? Uh, to add more complexity and really neat granularity to this, spectral cytometry became a really interesting tool that became available just when we were working on this project. And we asked at a single cell level, are these T cells responding differently in any ways in presence or absence of MHC class 2? So here what we are doing is pretty much repeating the same experimental timeline that we saw in the last slide, but instead of doing microscopy, what we are doing is doing spectral cytometry, and we had a 22 color panel, which was carefully selected to, number one, exclude the intravenous T cell so that we can really study what is truly in the lung. And it matters a lot for an organ like a lung because it carries so much blood because it has to oxygenate the blood. We included TRM markers like CD62L, 11A, and CD69. But most importantly, we included lineage-defining markers like TBET for TH1, GATA3 for TH2, ROR gamma T for TH17, and FOXP3 for Tregs. And the goal behind this was, like I mentioned, we know antigen presentation helps differentiation of the T cells. So we wanted to see, could these T cells be different if you knock out MHC class 2? With the help of Anna Belkina from the Boston University, we took all this multidimensional data and performed in silico data analysis and clustering to find that there were 21 different subclusters of these T cells at the protein level. I'm not going to talk about clustering and these things because eventually the functionality of the T cell is what matters. And there were two big surprises to be found. Number one, firstly, we found that there were T cells that were capable of expressing TBET and GATA3 and ROR gamma T, and sometimes even FOXP3 all simultaneously in the same T cell. So our immunology textbooks say that the T cell expresses one transcription factor and is one type of a T cell. Our model seems to suggest that that might not indeed be the case. The T cells might be way more multipotent than what we initially expected when we look at in a non-lymphoid tissue. And then another interesting observation we found is when we delete the epithelial MHC class 2, we see that the type of T cells that are TH1, 2, and 17 simultaneously were actually expanded in these lungs, suggesting that epithelial antigen presentation somehow is constraining the plasticity of these T cells. Another way to look at this really complicated data is essentially just make a simple pie chart where we put all the T cells that look similar together in the same plot. And like I showed you in the past slide, if you look at the lungs that do not have epithelial MHC class 2, these T cells that are TH1, 2, and 17 multipotent T cells are actually doubled in number, suggesting again the epithelial antigen presentation is somehow constraining this plasticity. Now, one might argue this might just be a figment of the fact that our mouse is a weird mouse because these are genetically engineered mice. So we took advantage of the fact that in the same mouse, we had given intravenous antibody injection, and we can go to the blood of that exact same mouse. And surprisingly, if we look in the blood of that same mice, this plasticity is nowhere to be seen. And this agrees with most of our literature, where we think that the T cells are largely monopotent or bipotent. However, only in a non-lymphoid tissue are we finding this multipotency. So hopefully from this work, one of the things I hope to convey is the conversation between T cells and epithelial cells is not a one-way street. In fact, in some spectral cytometry data that I have not talked about today, we also end up showing that different epithelial cells actually express different types of co-stimulatory molecules, and alveolar epithelial cells express a lot of PDL1. We go on to show in the paper that if you knock out the PDL1 by actually deleting, if you knock out PD1 on the T cells or the epithelial cells' ability to deliver the signal to the T cell, these T cells essentially lose this bottleneck or an evolve like constraint on them and start becoming very plastic and cause a lot of inflammation. So again, epithelial cells via PDL1 talk to PD1 on the T cells to essentially constrain their plasticity. So while we were working on all these questions, an important concern that started coming up in our heads was we breathe in a lot of opportunistic pathogens. Sure, I get it. But we probably breathe in a lot more potential allergens on a day-to-day basis. And our epithelial cells, at least in a healthy lung, somehow have to tolerate these allergens and not induce an allergic response in the lungs. So the question then became is, could there be a role for epithelial T cell interaction in context of allergy in this lung, in adult lungs? And staying true to our expertise with lung immunology, we decided to ask the question about allergy in the lung, which is asthma. And for the people who are not asthma experts, so asthma is a chronic inflammation of the airways, and it actually has two main features. First feature is the inflammation, where the epithelial cells and the immune cells, they sense these allergens, they get irritated by it, they produce a lot of chemokines to call for a bunch of immune cells like eosinophils and monocytes that cause a lot of remodeling in the lung. And this remodeling later essentially leads to what is causing the airway hyperreactivity, where an asthmatic cannot breathe because there is a lot of mucus that is plugging their airways. So inflammation and airway hyperreactivity are two very important symptoms of the disease, where it is believed that the inflammation usually comes first, and that leads to the airway hyperreactivity, which feeds the vicious cycle. And as the world is getting more and more modernized, there are more than 350 million people affected with asthma worldwide. So it is a very big socioeconomic burden. If I ask a layperson what type of asthma do they know of, one of the most common things people will think of is asthma has Th2 cells, produces a lot of eosinophils. And that is because we usually think of the asthma as more like an atopic, early life detectable asthma, or the early onset asthma that is highly eosinophilic. Why we know a lot about this type of asthma is that it has been very well modeled in the labs. So very old traditional models of asthma involve giving the mouse an model allergen like egg albumin in presence of an adjuvant like alum. And what this does, intraperitoneal injection does, is it induces Th2 cells against this allergen. And then we give the O albumin up down the nose, essentially inducing these T cells to come into the lungs and cause the phenotypes of allergy, which is associated with large amounts of eosinophils that you can see in the lungs and a large amount of Th2 recruitment that you see in the lungs. Such models have been very useful for us to understand very basic concepts and molecular mechanisms that drive asthma. And many of these molecules are currently targeted in the clinic with great success in helping treat eosinophilic asthma. In fact, if you go to the Global Initiative for Asthma recommendations, you can very clearly see many of these Th2 related discoveries that were made with mouse models are now targeted even in the clinic. So on the flip side, there is a whole different version of asthma called as neutrophilic asthma, which does not occur as frequently in young childhood, but occurs much later in adulthood. And it is severely driven by neutrophils. And one thing we know about neutrophils is neutrophils can cause a lot of damage to the tissue around them. Neutrophils are also steroid refractory, which means that this disease is actually irresponsive to steroids. And the worst part of this whole thing is that there have not been many good models to study neutrophilic asthma in mice. In humans, you can find Th1 cells and Th17 cells in these lungs. So the jury has been out if the Th1 cells are driving the disease or Th17 cells are driving the disease. And there are data suggesting both or one or the other are involved. And because there are no good models of asthma, that has also translated to the fact that the patients with steroid resistant asthma are actually very underserved. So if you look at the same plot from Gina that I showed in the past, you can see this tiny little box on the bottom, which says that if none of these things work, give an antibiotic like azithromycin, give a leukotriene receptor antagonist, and as a last resort, give oral corticosteroids and consider the side effects and hope for the best. And if you remember, I said this disease actually is steroid resistant as well. So many papers have ended up highlighting the fact that severe asthma, although it accounts for only 5 to 10% of the asthmatic population, the patients that suffer from that disease are paying for 60% of the asthma-related costs. So clearly there is some need to come up with a better model for this disease so that we can actually begin identifying some targets for therapy. As an immunologist, though, the question for me was, why have we failed to reliably model this disease? We have done so well with the Th2 high asthma, why not Th2 low asthma? And the reason for that is because a disease like neutrophilic asthma is calling for two different extremes of T cell phenotypes. You are asking for a Th17 driven neutrophilic inflammation while asking for airway hyperreactivity, which is driven by Th2 cells. And having both these type of inflammations in the same tissue in a mouse model has been a challenge. So how have immunologists been getting around it? Is what we do is we give adjuvants that push for neutrophils in the lung, something like CFA, which contains fragments of Mycobacterium tuberculosis, or something like cyclic di-AMP, which are bacterial PAMPs. And the idea is this would call for neutrophils under the fabric of a Th2 inflammation, and this would remodel asthma. And as Hamida Hamad and Bart Lambrecht's group very nicely put in their review in Cell, whether such models really represent type 2 asthma is still uncertain. So we said, coming from the world of microbiology and antimicrobial immunology, we came in from the notion that, look, we know that when we are born, our lungs are pretty simple and don't have a complex immune system, but microbial exposures as we are growing into adulthood induces a lot of T cells, of which I talked about, CD4 TRMs. And these complex immune response in the lung as an adult might be what helps us protect from lethal infections. Can this also be applicable for asthma, where early life allergen exposures might cause subclinical asthma? So it's not something that is going to really show itself as a severe asthma. However, over time, there might be remodeling in the lung that somehow breaks the camel's back, and that might lead to neutrophilic asthma, which also happens to be a late onset version and diagnosed later in life. So in other words, if we take this model of eosinophilic asthma, where we sensitize the mouse to O-albumin and expose them to albumin by nebulization, and instead of giving only one challenge, we let the mouse rest, give another challenge, and let the mouse rest, and then give another challenge, essentially to accumulate memory of their exposure to these allergens. And one of the things I will bring up here is every single mouse that you're going to see from here on has always been sensitized to O-albumin, which means that every single mouse has circulating T cells that are responsive to the allergens in these mice. The only thing that is different about these mice is some of them have gotten nebulized PBS, which means they have no lung history, which is why we call them the no LH mice, or they have nebulized O-albumin, which means they have a lung history, and equivalent to what we would think of as an adult lung, if you will. And to our surprise, within 8 hours of challenge with the O-albumin, we find that the LH mice seem to have a large amount of neutrophils coming into the lungs. We could find that the neutrophils were causing a lot of damage because of the amount of protein that was present in the lung that was leaking out of the vasculature. And more importantly, these mice, whether they be LH or no LH mice, still had the airway hyperreactivity visible in the lungs, suggesting that this model is actually mimicking many features of human neutrophilic asthma. In data I'm not showing you here, in the paper we ended up showing that this disease is steroid resistant, and the neutrophilic influx, unlike other models which peaks and then disappears, our model shows neutrophilic influx which is actually stable for quite a long time. So this truly does reflect a lot of features of human asthma. So if there are neutrophils in the lungs and there is experience in the lungs, we have already shown that Th17 cells can produce epithelial cells to induce CXCL5. So we asked, could this axis where the Th17 cell is talking to the epithelial cell to produce CXCL5 to call for neutrophil, if this axis is playing a role even in context of an allergic lung? And consistent with our hypothesis, yes, the experienced lung has a large amount of TRMs, produce a lot of IL-17, produce a lot of CXCL5, calls for a lot of neutrophils, which leads to a lot of damage in these lungs. What is interesting though is when we asked what type of T cells are producing this IL-17A, we performed again spectral cytometry to do some PMA ionomycin stimulations. What we found is, if you look at the experienced lungs, it is actually the CD4 resident memory T cells in the lungs that are by far the most dominant producers of IL-17 in the lungs. Again, suggesting that allergen experience induced Th17 T cells to produce IL-17 in these lungs. What is interesting about these T cells, unlike the previous T cells I talked about, was while they are producing copious amounts of IL-17, which is comparable to the IL-17 that would be otherwise produced by a antibacterial Th17 cell, they don't express that much ROR gamma T, which is the lineage-defining transcription factor of Th17 cells. Another way to look at the same data is if we put IL-17 on the x-axis and ROR gamma T on the y-axis, while the bacterial infection is inducing many of these double positive cells, which is what we classically think of as a Th17 cell, in context of our allergic lungs, there were these T cells that are producing IL-17 in absence of ROR gamma T, which was very surprising to us. We again asked if this is an artifact of our mouse model. So we decided to look at the single cell seq data set published by Dr. Sarah Teichmann's group looking at asthmatic lungs. And what we found here is if you look at the lung airway biopsy of human adult lungs, we could see ROR gamma T low IL-17 positive T cells in the cloud that marks TRMs based on their expression of CD69, 103, and the other markers that we commonly use to identify TRMs. We have been missing this T cell subset all this while because we just don't know what we should have been looking for. And our mouse model essentially gave us a window into the fact that, look, there is this whole subset of T cell that is expressing IL-17 in complete absence of ROR gamma T. So this was a really cool discovery that our mouse model allowed us to make. On the same side, if we look at the source of CXCL5, unlike in the context of pneumonia, where the alveolar type 2 cells were the main source of CXCL5, in our model, it was actually the mucus, so the mucin-producing airway epithelial cells were the main producers of CXCL5, which makes sense. Asthma is an airway disease, and mucus-producing cells are around the airways, right? Interestingly, if we look at antigen presentation signals, turns out these mucus-producing cells are also the most dominant antigen-presenting cells in context of a memory recall with the allergen. So what this suggests to us is, at least in an asthmatic lung, there are these micro niches that are essentially orchestrated by these mucus-producing cells that while they're producing mucus and causing asthma, they produce CXCL5 to call for neutrophils and talk to Th17 TRM cells to cause IL-17-driven inflammation. To see if this is indeed the case, we did microscopy on these lungs. And again, we find that in the same lungs, the neutrophils and CD4 T cells usually conglomerate around the airways, but the alveoli in this exact same mouse do not have these neutrophil influx visible, suggesting that, okay, these epithelial cells are what is orchestrating this influx of neutrophils and T cells and their conglomeration around the airways. We can also see clues for this in a human lung from Dr. Teichmann's work. where we find that the mucin producing epithelial cells in an asthmatic lung also happens to produce most of the CXCL6, which is the human ortholog of CXCL5, and also express bunch of MHC related molecules like MHC2, CD4 T, and some PDL1. So then the final obvious question is, if they are expressing MHC class 2, can we delete this MHC and what does that do? So we put our mouse through the same exact experiment where we deleted epithelial MHC class 2, put the mouse through the same, ushering it through the same protocol for inducing the asthma, and used the spectral cytometry panel to look at the T cells. And consistently in our model here again, we find that epithelial antigen presentation is required for helping Th1 and Th2 responses in an asthmatic lung. I would not make much out of the ROR gamma T signal here because remember our Th17 cells don't express the ROR gamma T, so we won't be able to catch them either ways in this experiment. However, considering the fact that there was low Th2 cells in the knockout, we asked if the airway hyperreactivity response in this lung was affected. And consistent with our hypothesis, in absence of epithelial MHC class 2, the airways are not resistant. That means they are not constricting as hard in the lungs, essentially meaning that epithelial MHC class 2 actually makes airway hyperreactivity worse. And this was a struggle for us. Why did we evolve expression of MHC class 2 on the epithelial cells if it is going to make breathing difficult for us, right? And here we have to remember asthma is again a bisymptomatic disease, one of which is airway hyperreactivity, but the other one is the inflammation. And when we looked at the inflammatory outcomes in these lungs by looking at the neutrophils, what we find is the deletion of MHC class 2 actually increases the amount of CXCL5 to call for more neutrophils and cause more damage in the lungs. So now it makes sense. Epithelial cells are trying to strike this very delicate balance of balancing Th2 and Th17 response, which historically have been difficult to model in the same mouse until some of these models like ours are allowing us to do so. Mechanistically, the question is then what is driving this increased CXCL5, right? And we assumed that it would be an increase in these Th17 cells, but it turned out to be a little more interesting than that. We did not find any differences in Th17 cells. Instead, we asked if epithelial MHC class 2 is helping Th1, could it be possible that the interferon gamma coming from the Th1 cell in retrograde is shutting this inflammatory response in the epithelial cells? In other words, could a classical pro-inflammatory cytokine like interferon gamma actually be inhibiting neutrophilic influx in context of an IL-17 inflammation? In vitro and in a simplistic model, that seems to be the case, where if we add interferon gamma, the amount of CXCL5 coming from the epithelial cell is reduced. In a mouse, the CXCL5 response is reduced. But then the true test for us was, if that is really the case, we should be able to supplement recombinant interferon gamma to these mice that have robust neutrophilic influx, and hopefully we can bring them down. And turns out that is indeed what happens. If we take the mice and give them recombinant interferon gamma, despite absence of epithelial MHC class 2, we could dampen the CXCL5 response and neutrophilic influx in these exact same lungs. So in this paper that we put out last year, what we are saying is that eosinophilic and neutrophilic asthma are not two different diseases. They can be diseases on a spectrum where the eosinophilic asthma can progress to become a neutrophilic disease. And clinically, this is not surprising to clinicians who see asthma patients, because there are patients that are initially responsive to steroids, but over time they become steroid irresponsive. However, the molecular and the cellular mechanisms as to what drives this switch was unclear. And our mouse actually is giving us a window into trying to see what is causing this. And what seems to be responsible, at least to some extent, is the fact that the epithelial cell via MHC class 2 is talking to the T cell and essentially setting this balance of inflammation and airway hyperreactivity at the same time. So with this, hopefully I have shown you, at least based on these three different vignettes, that epithelial cell and CD4 T cell interaction is actually very intimately married to each other, if you will, in context of microbial exposures or allergen exposures. However, one thing I do want to walk away from this talk emphasizing is expression of MHC class 2 or interaction with CD4 T cells is not a feature of only the lung epithelial cells. Over the past decade, there have been multiple papers, some of which I have put here as snapshots, showing that many different epithelial cells and many different barrier tissues are able to express MHC class 2 and talk to CD4 T cells. So, whatever functions we are looking at in the lung might have direct implications for discoveries that could be made at these other barrier sites like the gastrointestinal tract, the nasal epithelium, and the skin. So with that, I'll take some time to acknowledge a large village that requires to do work like this. So I'd like to thank Jay Misgard, with whom I did my postdoctoral fellowship in Boston, bunch of collaborators that helped us get these stories across the finish line, specifically thanking Anna Belkina, who's the director of the Flow Core, who played a very important role in helping me get started on using the SciTech Aurora and helping come up with complex flow cytometry panels and my funding sources. And in the recent years, since I've moved to University of Michigan, my current lab, including VJ, who was the main driver of that asthma paper that I talked about, the village of collaborators that have helped me set up the lab and helped me run the lab, the flow cytometry core here, the department here, and the funding sources.